Hello, testing my mic. Okay. So, okay, my mic is okay. Okay. Okay, so welcome. Good day everyone. Good evening everyone. So, now it's already 8:04. Okay. So we will start our class now. So today we will finish up the chapter 9, control and regulation, which will be the plant hormone topic. So last week I already covered the nervous system and also the animal hormone, the human hormone too, the mechanism about the steroid and non-steroid hormone. So today we will focus about the plant hormone. Okay. So in our syllabus, the plant hormone that we need to learn will be auxin, gibberellin, cytokinin, and also ethylene, and also abscisic acid. So there are 5 hormones you need to learn. So in SPM, you learn only 2 hormones, right? I think in SPM you only learn about auxin and also ethylene only. But in STPM, you need to learn about the 5 hormone. Okay, so the first hormone that you need to learn is auxin. So I think the auxin hormone is very famous for you guys, and also familiar to you guys lah, because you already learn in SPM too. So as you guys know, auxin is play the role in the growth. You know, like the auxin is the play major role in the growth, and there are 2 types of the mechanism that they can respond toward the light. Either it is respond toward the light or respond toward the gravity. So when the growth of the plant that respond toward the light, is phototropism, right? And then when the growth is toward the gravity, then we will call as geotropism. So normally auxin is produced in the apical meristematic plant. So if you still remember that in biology semester 1 that you learn about the meristematic tissue, we know that meristematic tissue is located at the bud, at the bud of the shoot or root, right? Okay. So normally auxin will be secreted at there. So there are no specific location. The specific location is only at apical meristematic cell only. And then normally it is transported from cell to cell, toward all the plant that require the growth of the plant. Okay. So normally this what I already mentioned about when the growth of the plant that using this hormone toward the light, we call as phototropism. And then we know that in auxin in shoot, the cells on the darker side will elongate faster than brighter side. Okay, so you will see this diagram. So normally this is phototropism because this is the plant growth of the plant toward the sunlight. So this is the shoot, apical meristematic of shoot. So we know that auxin is accumulated at here. So normally in sunny day, the auxin molecule will be accumulate at the darker side one. It would accumulate at darker side and elongate at the darker side part. So that is why that you can see that the plants are faced toward the sunlight. You see this. Why? Because the auxin is accumulated at the darker side and they will only elongate at there. The cells only elongate at the darker side. Darker side, the cells does not elongate. It only maintain at there. But only this one, the cells are elongating. So that is why that you can see that why the plants will face toward the sunlight. So this is the reason auxin. And another one is about the geotropism. So there are 2 types of geotropism, but positive and negative. So normally if you still remember that geotropism is the response of the auxin toward the growth of the plant based on the gravity. So you see here the auxin is accumulated either in shoot and root, right? So in root, especially in root, the Auxin will face toward the gravity. It will accumulate based on the gravity. So in root, it will accumulate at the lower part only, similar as the shoot. It will accumulate in the under layer of the shoot only. But the difference is auxin stimulate growth in the shoot. So when the auxin is accumulated at the under layer of the shoot, it will stimulate the growth. But in root, the auxin function opposite way. It will inhibit the growth. So meaning that only the upper part of the root will be growth only. The upper part will be elongated only. So it is totally two different function. Auxin in shoot is stimulate the growth, but auxin in root will inhibit the growth. So when you talk about the function of auxin, we normally talk about the shoot only, because root will inhibit the growth one. Okay, so this is very important that you need to know. Okay, and then you still remember that I just recap from the SPM. I think SPM you already learned about the experiments that prove about the relating about the auxin molecule. So normally one of the experiment is conducted by the Charles Darwin. So normally this experiment is to determine phototropism of the auxin. Whether why we say that the auxin will be faced toward the sunlight one. Why? So normally these scientists use the five parameters to conduct the experiment. So the first experiment is the control meaning that all the plant shoots are not be cut down, or we call as collotype. This one is collotype. So meaning that the collotype is maintaining. We act as control we not change any part of the plant structure at there. But in second diagram, we remove the collotype. Okay. Once we remove the collotype, they found that actually no any response at all, meaning the shoot also cannot grow. No shoot will be growth. The shoot is maintaining at here only. Not meaning that the plant is die, but the plant cannot grow. It only can maintain the height of the shoot like this only. So this meaning that auxin is highly concentrated in the tips of the collotype, or in the tips of the shoot. So if you cut down the tips of the shoot, then no auxin produced, so that the plant cannot grow anymore. And then you see this diagram. When you cover the collotype with the cap, or we known as the opercle cap, meaning that this is totally black cap. No untransparent cap. It is totally dark cap that can totally inhibit the light enter into the plant. So that since there are no plant, there are no plant, no sunlight at there, so there will also no any response to, because auxin only respond when they are sensitive to the light only. So if no light response, the shoot also cannot grow. But when you cover with the transparent cap, meaning that the light still can pass through the cap into the collotype, so that the auxin can be will have a response. So the auxin will like the positive tropism. It will accumulate at the darker side, and then promoting the cell elongation at the darker side. And then the fifth diagram is they cover at the base of at the stem of the plant, rather than on tips of the plant. So when they cover in the on the stem of the plant, they found that the response still occur, similar to the transparent cap, totally similar. So meaning that there are still the plant light response toward the by the auxin, which the plant face toward the light. So they are still respond by the auxin too. So in this experiment, they conclude that auxin can be found in the tips of the shoot only. It only can be found on the tips of the shoot only. Okay, and then when and the first conclusion, and then the second conclusion is when although you remove the all the tips of the shoot, you still tip of the shoot, but no light toward the auxin. The auxin still cannot have a light response for phototropism. So this is the conclusion that concluded by the Charles Darwin about the phototropism of light response from auxin. Another experiment is by Jensen. Okay, so normally this one is they using the any barrier that can be cut, that can be inserted unilaterally, or using the gelatin barrier inserted subapically. So you will see this diagram, meaning that they inserted this mica barrier is directly inserted into the tips of the plant, but only half only, not totally cover the plant, but only half. So you see here when this arrow is indicating the light response. So you see here when there are the barrier at here, meaning that when the light is come from the right hand side, there are no response at here. And then if you see this, when the barrier is inserted at the right hand side, and then the light is come from right hand side, but there are response at here. The results show that there are response at here. So meaning that auxin only can accumulate at the darker side and provide elongation toward the light response. But for this one, since it is already barrier at here, they already got barrier at the darker side, so that auxin cannot accumulate at here, so no light response by auxin. And then this one is they use the gelatin barrier. So normally this one is they totally cut, and then like previously experiment, if you totally cut, they will totally cut, and then they cover by the gelatin barrier. So this one is inserted, they just cut the portion of the tips of the plant, and then they insert half only. But this one, they totally cut down the tips, and then they insert the gelatin barrier. Okay, after that, they found that they are response. They will have a response toward the light. Meaning that if you using the gelatin molecule, meaning that the auxin still can accumulate at the darker side, and then still stimulate the growth of the plant, compared to using mica barrier. If you're totally using mica barrier, meaning that mica is the totally blocked, right? You totally block it, and then no response at all. Okay. Another experiment is by Vance experiment. Okay, so this experiment is where they using totally cut down the tips of the plant, and then they transfer the auxin to the jelly. And then later that they'll transfer this jelly already high in concentration of auxin, transfer back to the plant again. So they transfer back to the plant again without the original tips, but using the agar block that containing the auxin serum. So after they transfer back again, they found that Auxin block cause bending of shoot not exposed to light. So meaning that the hormone from tip is stored in agar block, and then the agar block put on colluted on the side, and then the colluted will bend toward the light, and then auxin move down the shaded region for response to growth. So they found that either you using the right hand side or left hand side, you cover the right hand side and left hand side, but if they are the light, they still can respond to the light. Cells on the shaded side elongate in response to the hormone. Okay. And another one is experiment by Bridge. Okay, so normally this one is they are using the similar, they just modify the experiment by veins, but the difference is the auxin tips that the shoot tips that you use is they were extracted with the light first, and then they they use the barrier to block the auxin development. Okay, so meaning that if you see here, you see this diagram. So when the barrier is auxin blocked, so when they are the light from the left hand side, so if you still remember that auxin will be accumulate at the darker side one. So meaning that they will only accumulate at the big B part B only. The auxin will be accumulate at the part B region only. So meaning that part A region is less concentration of auxin sebum. So when you use this agar block, transfer this agar block toward the plant again, you will find that the curvature of the plant for A and B are different. B will curvature more curve, and then A the curvature is very low. The degree of angle of curvature is low, but for this one the degree of angle is high. So why? Because B containing high concentration of auxin, so it will more highly respond toward the light compared to A. Okay, so this is the bridge experiment that want to tell you that the light response of the auxin. So even though in the agar block not whole plant, but the auxin will respond only in the darker side, not at the light side. Okay, so this is all the experiment that related to auxin lah. So although syllabus does not mention, but you should know too, because in SPA you already learned. So whatever maybe in form 6 and then they come to tell you. Okay, now we go to function of auxin. So this is very important part. What about the function of auxin? So the primary function that I already mentioned before, cell elongation, primary growth. Primary growth is the main function for auxin. So we know that cell elongation meaning that it is to elongate the plant cell. It is not a growth, not cell division. It will not produce many plant cell, but it provide the larger plant cell. So that is why we call as cell elongate. It elongate the plant cell. So normally this is the response of the auxin toward the cell elongation. So normally in cell elongation, the most challenging we want to elongate the cell actually is because of the component of the cell wall. We know that cell wall is very rigid, and then it protect the plant cell. So you want to elongate the plant cell is not very easily. You need to softening the cell wall first. You need to loosening the cell wall structure first. So how? The auxin firstly will increase the activity of proton pump, which hydrogen ion from the cytoplasm. So the hydrogen ion will reduce the pH of the cell wall. So meaning that the auxin molecule will increase the proton pump from the cytoplasm, meaning that it will pump more hydrogen ion into the cell wall from the cytoplasm, so that it will reduce the pH of the cell wall, meaning that the cell wall is more acidic lah. So since the cell wall is acidic, it will activate the protein called expendencin, red color one, expendencin. This protein will separate the microfiber from the polysaccharide. We know that the structure of the cell wall is composed of cellulose, right? Cellulose, one type of polysaccharide, and then not only cellulose, you have lectin molecule, phycofiber, hemicellulose, lignin, all these. But this one is only primary growth, so the structure is not very complicated compared to the woody plant. So normally normally they are the microfiber which will give the shape and protect the plant cell wall. So normally microfiber will be separated from the cellulose molecule. And then this polysaccharide, so since the microfiber is already released, microfiber and the polysaccharide are separated, meaning that the loosening of the cell wall is occur now. So the polysaccharide will be degraded by using the enzyme. So this process loosen the microfiber, making the cell wall more flexible. And then water uptake and increase sugar then cause the cell to elongate. So you want to increase the size of the plant cell, you need to make sure that the cell wall is flexible. If not, if your cell wall is too hardening, you will still elongate the cell will burst, right? Your cell will burst. So you need to loosening the cell wall first by separating the microfiber and cellulose, so that the polysaccharide can be degraded, and then the microfiber can be loosened, and then making the cell wall more flexible, so that the water molecule come into the plant cell can fill up the vacuum, so that it can increase its size. So this is the major function of auxin. Okay, so this is the explanation. Okay, so you can read yourself. Okay. So another auxin function is it enhance the apical dominance. Okay, so firstly you need to know what is the meaning of apical dominance. So apical dominance is the inhibition of growth of axillary buds by the apical bud of a plant shoot, meaning that apical dominance is at here. Apical bud is located at the tips of the plant the tips of the shoot plant. Okay, and then if apical dominance is occur, meaning that the plant will not, dia tak akan cabang. The plant will only grow in regular pathway. You see this, it will grow in regular one. Dia tak macam ni, you will see this, dia akan cabang-cabang ke sana, dia akan cabang ke sini, cabang ke sana. So this one is not apical dominance lah. It already is already about the non-apical dominance already. But apical dominance is like this one. It look like the regular shape, and then more nicely lah, and arrangement is more nicely and more regular compared to this one. So this one is already without the apical bud. So apical bud is, we know that apical bud is the place that to secrete high amount of auxin. So the axillary bud remain dormant unless the apical bud is cut. So the axillary bud, you will see this axillary bud at here, ini, for this is remain dormant, meaning that they will not grow, tak akan cabang-cabang macam ni punya. And then if the apical bud is cut, the axillary bud will grow and take over as the new apical bud. So if you cut down the apical bud like that, it will totally cut down, so no auxin in the plant, in the shoot of the plant. So the axillary bud will grow. So that it will look like this. So you see this diagram, the apical bud is a preferred sugar sink and major site of auxin biosynthesis. So the upper part, this upper part is we call as apical bud. So auxin will move downward from the apical bud to replace the growth of the axillary bud. So all these axillary bud here, you see this is we call as axillary bud, are remain dormant because got auxin. So it will not grow. The apical bud will not grow. Not apical bud, axillary bud, sorry. Okay, so axillary bud. And then there are another hormone is called as cytokinin. Later I will explain it. But for now you need to know that auxin is the play major role in enhanced apical dominance, where it inhibit the growth of the axillary bud. Okay. And then there are also other functions. So normally also Auxin also can alter the gene expression, causing cell elongation to produce new protein. And then also promote food growth and induce paternocopy without fertilization. And then Auxin can also inhibit abscission process to maintaining the structure of the cell wall. And then Auxin can also play the role establishing phyllotaxy and arrangement of leaf on stem lah. And then also Auxin can promote cell division in vascular cambium, leading to secondary growth. So these are all the functions that you need to know in Auxin molecule. Okay. Okay, next hormone is about cytokinin. Okay. Okay, so normally cytokinin is synthesized in the root and transported to other organs, as although there are many minor sites of production as well. So the major synthesis site of the production of gibberellin is root. Compared to Auxin, Auxin is meristematic cell at the root and shoot. For cytokinin, it is produced in root, but actually other part also can produce as well, but the most important one is at root. The major biosynthesis of the gibberellin cytokinin is in root. Okay, and then this is the general function. So normally the main function is to regulate cell division in shoot and root. Yeah, just now Auxin is play the role in cell elongation, but cytokinin play the role in cell division. These are totally two different functions. Cell division and cell elongation are different. Cell elongation just increase the size of the cell only. It will not divide. It will not cannot produce 2 cell, 3 cell, 4 cell like that. It will not beranak. In human, you can say it tak akan lahir lah. Dia lahir anak-anak dia. But cytokinin, it will lahir anak-anak mereka. And then not only that, it will also modify apical dominance, promote lateral bud growth, and promote movement of nutrient into zinc tissue, stimulate seed germination, and also delay leaf census. But mostly we will focus about cell division and also the apical dominance. Okay. So this is the roles of the cytokinin. So it is act as the control of cell division and differentiation process. So normally cytokinin will be add in with the Auxin one. It will like the abang-adik lah. Kalau ada Auxin, memang ada cytokinin. They will collaborate to each other one. So cytokinin stimulates cell division and influence the pathway of differentiation. So normally when you have the tissue, when the plant cell is cultured in absence of cytokinin, the cell will go very large but does not undergo mitosis. So this is the key point that I want to tell you. Meaning that Auxin is only grow very large only. The cell number is still maintaining what you place. How many cell number in the culture, meaning maybe 10 cell, you place 10 cell, then finally you only have 10 cell too. But thus the cell size will very large only. But cytokinin will play the role in mitosis, cell division. It will produce large number of cells. So maybe you culture 10 cell, in maybe 1 week, 2 weeks, you will have 20 cell, 30 cell, or even 50 cell like that. Okay, so normally cytokinin can collaborate with the Auxin. So the ratio of cytokinin and Auxin can control the cell differentiation. So you will see this diagram lah. So this diagram I summarize into this lah. So when you Auxin is equal to cytokinin, meaning that it will induce catalyst. So what is catalyst? So catalyst actually is the plant is you can say that is one type of the plant structure that can play the role in further cell development. Meaning so in general, the plant in natural habitat, plant will not induce catalyst one. Normally this one is that human made in tissue culture technology. Normally this one is because of the human want to do the experiment about the plant. So if you want to do the experiment in the plant, normally we will not grow the plant as well like the farmer. In laboratory we will induce the catalyst only, because this catalyst later can develop into leaf, can develop into shoot, can develop into and flower, like that. All the plant structure can be developed from the catalyst. So it is very useful structure for the researcher to make a research about the plant tissue culture lah. So so in natural, catalyst will not form in natural lah, very rare. It is very rare situation that the plant will form catalyst. But for generally, plant will either that the auxin concentration is higher than cytokinin, or cytokinin is higher than auxin. So when auxin is higher than cytokinin, it will induce rooting. So meaning that when the plants are exposed more auxin concentration, it will induce rooting one. Only auxin induce rooting. But if cytokinin is higher compared to auxin, it will induce shooting. So induce rooting mean that the growth of the shoot is very slow, or even no growth, but the root will grow very fast. Opposite to cytokinin, when cytokinin is higher than auxin, it will induce shooting, meaning that the root will the growth of the root is very slow, but the shoot growth will very high. So this is the meaning lah about the cytokinin. Okay, and then it also play the role control of apical dominance. So as I mentioned before, we know that auxin play the role in enhance apical dominance, and we know that apical dominance it inhibit the growth of the axillary bud. So when you remove the apical bud, no auxin at all, cytokinin from the shoot antagonize the action of auxin, allowing a limited amount of axillary bud growth. So it will induce the growth of axillary bud. So the axillary bud farthest from the apex are increasingly elongated. So meaning that if although apical bud is still maintaining at there, meaning that got auxin lah. Although you have the plant have the auxin yet, but the auxin but the cytokinin still can antagonize, it will counteract the action of the auxin one. It will still can stimulate the growth of axillary bud, but the growth is very slow, not very fast like this. This one is where the auxin is already totally removed, meaning that auxin level also decline, particularly near the gut surface, and then the decline topmost axillary bud in particular to go and take over the new apical bud. Yeah, so I just want to mention about the cytokinin play control about the apical dominance. It can control the apical dominance one. So yeah, like that. Okay. And then another function is about anti-aging effect. So meaning that anti-aging, we know that aging is tua, right? Orang itu bahasa Melayu kami panggil orang tua. We know that in human we will have going to death phase lah. Kami akan jadi tua kan, 60 tahun, 70 tahun akan jadi tua, lepas tu mati. But cytokinin can prevent the plant go to aging. So that is why some of the plant why the plant can grow unlimited, because got cytokinin. Cytokinin already play the role in cell division, and then it also can provide anti-aging effect. So that is why some of the plant cannot die one, and then secondary plant it will have the unlimited growth. Okay, so cytokinin slow the aging of certain plant organ by inhibiting protein breakdown. be brought down. And then if leaf removed from a plant are deep cytokinin solution, they stay green much longer than otherwise. So this is the role of cytokinin lah. Okay, next hormone is about gibberellin. Okay. So normally gibberellin will be the site production is in the apical bud and also root, young leaf and developing seeds. So there are many site production lah. Apical bud. So this apical bud, and then not only apical bud, root, and then young leaf and also developing seed are the primary site of the production. So normally these are the general function. It can stimulate stem elongation, pollen development, pollen tube growth, fruit growth, seed development, germination, regulate sex determination, and transition from juvenile to adult phase. So normally you see this function, this hormone, it more responsible to sexual regulation. What? Stem elongation is not, but pollen development, pollen tube growth, fruit growth, seed development, all of these are involved in sexual reproduction. But so normally this hormone play the role in sexual reproduction, especially in seed germination. So one of the major function of the gibberellin is stem elongation. So you see this diagram. So this one is no gibberellin. This one we use gibberellin. So are best known for stimulating stem and leaf growth by enhancing cell elongation and cell division. So it will activate the enzyme that loosen the cell wall and facilitating entry of expansive protein. So normally, if you compare to Auxin also play the role in cell elongation. But Auxin is found in majorly found in the bud of the plant. Either it accumulate at the bud of the shoot or accumulate at the root bud, meristematic cell. But in gibberellin, it provides the stem elongation. So you see this diagram. If only the plant only containing Auxin does not have the gibberellin, the plant still grows, but only the leaf only can grow. The only leaf can grow, but the height of the plant cannot be increased because no gibberellin. But if you have gibberellin, it will induce the growth of the stem so that the stem, the height of the plant can be increased because all this stem is elongated by gibberellin. Okay. And then also gibberellin also play the role in fruit growth. So the very special case that not only ethylene, we know that ethylene is provide the ripening of fruit, right? But gibberellin is provide fruit growth. This is not the fruit ripening process, it is a fruit growth process, meaning that fruit is already ripened, but the fruit still need to grow. So gibberellin play this role. So this is a very common important application in gibberellin is spraying the grape. So normally gibberellin they will play the role. The one of the application in the gibberellin is the fruit that never mature fruit. They will cut down from the trees, and then they will pass to the industry or go to the supermarket, and then after they reach the supermarket, they will spray the gibberellin to stimulate the mature of the fruit. So yeah, so this is one of the application that you need to know. And then the gibberellin also make the internal of the grape bunch elongate. Okay, so this is one of the function. Okay, another function is about seed germination. Okay, so seed germination I hold on first because this one you will learn in chapter 4. There are one topic, the special topic about the seed germination. So gibberellin play the role in seed germination. Okay, so this one I hold on first. I will teach you next chapter. Okay, the fourth hormone that you need to learn is about abscisic acid. Okay, so abscisic acid is totally a reversible function compared to the first three hormones, auxin, gibberellin, and also cytokinin, because abscisic acids play the role in inhibition growth. The first three, all them are promoting growth. Auxin promoting cell elongation, but it only inhibit growth in root only, right? It only inhibit the growth in root only. But still, the main function is to provide cell elongation. And then you know that cytokinin is play the role in cell division and also cell differentiation. And then we know that gibberellin play the role in stem elongation. All of them promoting growth of the plant. But abscisic acid is inhibit growth of the plant, totally reversible. So if you see the site production of the abscisic acid, almost all plant cells able to synthesize. So there are no major site production. Almost all the plant cells, if you are the plant cell, they were able to synthesize, secrete abscisic acid. But normally, okay. And then the major function is inhibit growth. So there are many functions to inhibit growth lah. So normally stomata close, you close your stomata, seed dormancy, inhibit germination, and then promote leaf census and promote desiccation tolerance. Okay, so we see one by one. So one of the role is seed dormancy, meaning that it maintaining the shape of the seed. It will not make the seed germinate. You know that gibberellin promotes seed germination. So this abscisic acid will provide the reversible function. It will dominate seed dormancy, meaning that it maintain the seed shape. It will not the seed will not germinating. Okay, so normally seed dormancy. So normally in the seed, it will have a higher amount of abscisic acid lah. Okay, so maturing seed inhibit germination and then induce the production of protein that helps seed withstand the extreme dehydration that accompany maturation. Okay, and then many seeds have high level of abscisic acid. So ABA is a short form of abscisic acid in their tissue and do not germinate until the abscisic acid are washed out. Okay. Another one is drought tolerance. So this one is especially in the hot sunny day. Normally in desert lah, or when the place like now, maybe in few days ago, when the when the weather is very extremely weather, meaning that there are no raining at all. Meaning maybe 1 week, 2 weeks, no raining at all. So that this is what we call as drought tolerance. It can tolerate the drought, the drought condition. So meaning that when a plant begin to wilt, so normally when the plant is begin to die lah, already lack of water, tak ada air lah. Kalau tak ada air, ABA abscisic acid will start to accumulate in the leaf and close the stomata rapidly. Although in sunny day, you still remember in chapter 7, you still remember that opening of stomata is due to the sunny day because of blue light, right? Blue light to stimulate the bottom pump, and they cause the stomata to open. But for the when during the drought condition, there are no water as well. Dah banyak hari tak ada air, so that the abscisic acid will directly close the stomata to reduce the transpiration rate. Okay, so normally how the mechanism, so the simple mechanism is it will affecting the second messenger. So the ABA will cause the potassium channel in the gut cell to open. So it will miss lazy loss of potassium ion, so that the osmotic loss of water will reduce the gut cell trigger and lead to closing of stomata. So this is like the closing mechanism. So this is very important. Maybe come out in essay question, especially in drought condition. So this meaning that although in sunny day, there are the certain unique condition when the plant is begin to wilt or under the drought condition, so that the plant, the stomata will also close. So this is the mechanism lah. Okay. So the last hormone, ethylene, plant hormone ethylene. Ethylene is, you still remember in SPM, you know that it is promote food ripening, right? Yeah, this is a major function of the food of the ethylene. But not only food ripening, but they are also the other function too. So normally ethylene is the gas hormone compared to other type of hormone. All the other type of hormone is normally is liquid hormone. But for this ethylene, it is a gas hormone and produce almost all the plant, and then high concentration during senses, lip excision, and also food. When they are containing food, you will have the high concentration. So normally, the main function is to promote food ripening, lip excision, and then trigger triple response, and then enhance rate of senses, promote root and root hair formation, root hair formation, and promote flowering. So we will sing one by one. So one of the main function, food ripening lah. So I think I don't need to mention well. So normally at ethylene can break down the cell wall of the cell wall on the food to softening the food. So that and also it will also can break down the starch and acid to become sugar to make the food is sweet. So that is why that when you taste the food, you taste sweet lah, because of this. And then the production and color help advertise ripening process to ripen to even animal which eat the food and disperse the seed. So and then ethylene will trigger ripening, and then the ripening will trigger more ethylene production. So this will result a huge burst in ethylene production. So you need to know that ethylene is gas hormone. So when they are trigger ripening and the foods are ripened, already ripened, so that this food will also trigger more ethylene too. So I already mentioned it is a gas, so the distance will spread from food to food. So not only the food ripening, they are also respond to triple response to mechanical stress. So what are the triple response? So I already mentioned triple response. So there are three main response lah. So normally this hormone ethylene also can instigate a growth maneuver known as the triple response that enable to shoot to avoid the obstacle. So meaning that ethylene when the plant is groove, when they are the obstacle at there, meaning that if let's say there are the barrier at the head here, so the plant cannot go up already, because in general condition it like this one, it will go up. But when there are the obstacle at the top of the plant need to avoid this obstacle, but it still can grow. So this ethylene will respond to mechanical stress. So there are three main response. So one of the first response is to slow down the stem elongation. So you see this, when there are the ethylene gas at here, when you promote the ethylene gas to this plant, you found that the growth of the plant will become curve shaped, right? The stem will become more curved, so that the stem elongate slow. So it will thickening the stem, make the stem more stronger. So you see that the thickness of the stem will become more thicker, and then the curvature that cause the stem to start growing horizontally. So as the effect of the initial ethylene pulse lessen, the stem will resuming the vertical growth. So meaning that when the already pass through this obstacle, actually it will continue with vertical growth. But when it contact a barrier again, so the ethylene will start to release again and continue the horizontal growth again. So there are the three main response. First response is to slow down the stem elongation. Second response is to thickening the stem, and then the third response is curvature of the stem. And then another one is senescence. So what is senescence? So the meaning of senescence is programmed death of certain cells of organ of the entire plant. So meaning that when the plants are die, when plants also like human, they are also organism. So the cells or the plant organ may be like may also will die. It will not have a unlimited lifespan one. It will also have a cells like an organ will be die in the plant one. Okay, so normally cells, organ and plant genetically programmed to die on a schedule do not simply shut down cellular machinery and await death. So compared to human, it will not completely simply shut down the cellular metabolism, and then just tunggu mati je. Tak ada punya. Dia ada program dia. So normally at a molecular level, onset of program cell death is a very busy time in a cell life require a new gene expression. Okay, so normally there are the many enzyme form to break down the chemical component, including chlorophyll, DNA, RNA. You need to break down all the component inside the cell inside the cell. Meaning that the cell does not like a human die lah, mati je lah. Tak, dia kena, you need to break down one by one first. Okay, so the plant will salvage many of the breakdown product, and then burst ethylene is almost always associated with the death of the cell during census. Okay, the another function, last function is about the leaf excision. Okay, so normally leaf excision is the loop of the leaf. Loose of the leaf from deciduous from the tree that help prevent desiccation during seasonal period where the availability of the water to the root is severely limited. Yeah, so meaning that this one is kejatuhan daun lah. Kejatuhan daun. Okay, when an atom leaf fall, it detached from the stem at an abscission layer that developed near the base of the petiole. So you see this diagram. So there are the two layers, right? Protective layer and also abscission layer. So normally the small parenchyma cell of this layer have very thin wall, and there are no fiber cell around the vascular tissue. So the abscission layer is further weakened when the enzyme hydrolyze polysaccharide finally the weight of the leaf with the help of the wind and the separation within the excision layer. So this one is play the role in about the leaf excision, meaning kejatuhan daun lah, daun akan jatuh. Okay. And then also there are also other type of plant hormone, but this one is not under your syllabus, so I don't focus lah. If you are interesting, you can read yourself. Okay. Okay, so I think that's all for the plant hormone. So I need to see any questions so far. Okay. Okay, if no question, I will proceed to the photoperiodism, the last topic for plant hormone topic. Okay. Okay, let me stop sharing first. Okay. Okay, now we will proceed to the last topic of this chapter, photoperiodism. So this is a final topic about under the plant hormone part lah. Okay, so before we go to photoperiodism, you need to know about the cytochrome first. So in our plant, there are also there are many pigments. The one of the pigment that you already learned is chlorophyll, right? Chlorophyll pigment. They will absorb the light, the blue light and the red light one, to induce the photosynthesis process. But not only chlorophyll pigment, there are also another pigment we call as phytochrome. Okay, this phytochrome actually is respond to the light too. It will respond to the light. So normally it will either absorb the red light and far red light. There are two types of light you need to remember: red light and far red light. To regulate many plant respond toward the light, including seed germination and shade avoidance, and also flowering process. So I need to explain about the red light and far red light, the mechanism between the cytochrome lah. So you see this, using this diagram, you see that PR is red light and PFR is far red light. Okay, so normally PR absorb red light in the maximum wavelength of 660, and then PFR, the maximum wavelength is 730 nanometer. And then during the light, we know that in sunny day, you still remember that you learn about the chlorophyll pigment. You remember that during the sunny day, there are two light that are highly found from the sunlight, which are what? Red light and also blue light, right? Red light and blue light. So during the day, meaning that PFR were higher than PR. Far red light is highly concentrated to red light. Why? Because during the sunny day, red light PR will be converted to PFR using the red light. So meaning that when there are the red light respond toward the plant, PR akan tukar kepada PFR. So that is why that PFR akan sangat tinggi during the sunny day. Kerana semua PR ni dah tukar kepada PFR lah. So red light increase the PFR, but in vice versa for night. So during the malam, during the night, PR were higher than PFR. Why? Because during the night, red light is limited. No red light at all. No sunlight already. So no sunlight equal to no red light. So there are only far red light. So far red light is during the night. So during the night, all the far red light, PFR, cytochrome far red light will be converted into PR. So this is the very conversion process. PR akan tukar kepada PFR when they exposed to red light. So that is why that we call as PR, kerana dia absorb red light. Lepas tu dia akan tukar kepada PFR. And then PFR akan absorb far red light during the night, tukar kepada PR, red light, PR. So normally this process, it will inhibit the response to seed germination. So normally red light will increase the percentage of seed germination, meaning that PFR is the form of phytochrome to trigger the development of the leading to seed germination. So PFR actually can seed germination, inhibit of vertical growth, stimulate the bunching, and also setting the internal control of flowering. Okay, next is about the photoperiodism. So normally this one is photoperiodism is highly related to the cytochrome because it is a response toward the specific night or day length, such as flowering. Now meaning that in one plant when they want to provide a response, you will see this one is based on the concentration or the number of cytochrome only. So if your PFR is high, so you will initiate seed germination. But for this one, there is the physiological response to the night or day length, meaning that when the plant only will respond towards something when the night is reach the certain length. So like a flowering. So in our syllabus, we only focus on flowering process only. Flowering. So meaning that bila plant tu flower, bila tumbuhan tu berbunga. Kami tahu dah durian, pokok durian. We know that durian trees will only flower between June, July, and August only, right? Others period will not flowering. The durian tree will not flower. Why? Why only durian can flower during June, July, and August only? Why they cannot flower during January? Kenapa? Ada sebab dia. So normally in our syllabus, we focus on flowering based on the night or day length. So there are the two types of plant: long day plant and also short day plant. Okay, so long day plant are the meaning that the plant or this plant only will flower when the day length, the day length meaning the sunny day lah. The day length is longer than critical point. So there are the critical point at there, critical dark period. So normally critical dark period is on 12 hour lah. Either 12 hour lah. So meaning that in one day, when the day is more than 12 hour, meaning that the long day plant will flower. Conversely to short day plant, when the day is less than 12 hour, maybe only 8 hour, only 7 hour like that, it will flower. So this is two different things. Long day plant only will flower when the day is longer than critical point. In short day, the plant only will flower when the day length is shorter than critical dark period. So these are all, and then the above I take example. So normally the long day plant example includes tomato, rice, and dandelion. But actually there are more examples lah. But tomato, rice, and dandelion are the most famous. And then short day plant including soybean, poinsettias, and also chrysanthemums. Okay, so normally you need to know also the mechanism how the plant flower. So I already mentioned in short day plant, the plant only the flower when the daylight period is shorter than critical length. So meaning that another meaning is the dark period is longer than light period. So your PR level increase and your PFR level decrease. Right. In dark period, you still remember your PR increase because far red light is high. So all your PFR will absorb far red light and convert to PR. So that is why that your PR will increase. So that your high level of PR in short day plant will stimulate the floragen. So floragen is the key hormone, what we call as flowering hormone, to promote flowering. Okay, so that the flowering hormone acts as a floragen hormone acts as a flowering hormone to induce flowering in short day plant. Okay, in long day plant, I already mentioned it only flower when the light period longer than critical length. So another meaning that when your dark period is shorter than light period, meaning that your PR level is decreased, right? Because your sunny day, your daylight is containing high amount of red light. So if you containing high amount of red light, all the PR will be absorb the red light to convert to become PFR. So high level of PR in long day plant will stimulate the inactivate floragen. So similar to that. So high PFR in long day plant will stimulate inactivate floragen hormone become activate, and then this hormone will stimulate the flowering lah, so that the plant can flower. Okay, so this is the mechanism between the short day and long day. Okay, so I think that's all for today. So I think that's all for today class. So any questions so far? No question? One. Okay, so I think most of you know question. So today I already finished up the chapter 9 lah. So tomorrow we will enter chapter 10, reproduction, development and growth. Okay, so that's all for today class. So see you guys tomorrow, and have a good rest and have a nice day, and see you guys. Bye.